Welcome to studyhigh.pk. Today I am going to start a new chapter of MBBS Biochemistry, chapter Bioenergetics lecture 1. In this lecture, we will discuss Gibbs free energy and I will explain in detail why we call it Gibbs or free energy. What is the significance of understanding this topic in comprehension of different types of chemical reactions happening in living organisms, and our second topic of discussion is standard Gibbs free energy. What is the difference between the Gibbs free energy and standard Gibbs free energy? Standard Gibbs free energy is measured as standard state, and we will also discuss what is the concept of standard state or how the standard states of biochemical reactions is different than that of other physical reactions happening in your chemistry lab. We will also calculate standard Gibbs free energy from Gibbs free energy formulas, and another important thing that I will explain is the relation of Gibbs free energy with second law of thermodynamics. Second law of thermodynamics uses a concept of entropy, and this concept of entropy will also help you in understanding the Gibbs free energy. So let's start our today's lecture. First of all, we will discuss what is the Gibbs free energy. In 1870, William Gibbs uses the word available energy for some biochemical reactions that are happening in human body. So William Gibbs, like in our body cells, in cytoplasm glycolysis occurs, and during different steps of glycolysis, like just take an example, when phosphoenol pyruvate converted into pyruvate, the Gibbs free energy or simply Gibbs energy for this reaction is minus 61.9 kilojoule per mole. And a part of this energy, approximately 30 kilojoule, is used in formation of ATP, and other remaining energy is used or excreted in the form of heat out of the body and or for driving different types of chemical reactions. So what will happen? There is some available energy in this reaction that can be used in formation of ATP. So Gibbs uses the word available energy for some chemical reactions. So this word Gibbs is also after scientists. Free, do not be confused by the word free. This is just used by the scientist Gibbs, whereas some scientists do not use the word free. They just say it gives energy. Like when the first step of glycolysis, during the first step of glycolysis, what happens? Glucose plus phosphate combine to form glucose 6-phosphate, and energy or Gibbs energy for this reaction is plus 13.6. This is an endergonic reaction. So not all reactions provide you available energy. Some reactions will require energy for their completion. So we cannot say that always free. So you can also say Gibbs energy. So this is the definition. It is that portion of total energy that is available for doing work. So let's move forward. You also know, or you must have to know, there are three types of conditions. One are exergonic reactions, like I have explained you the how when phosphoenol pyruvate converted into enol pyruvate or pyruvate. In exergonic reactions, delta G, Gibbs free energy is represented by delta G, is negative. It means that during this chemical reaction, or for those chemical reactions for which delta G is negative, they are spontaneous and they will release some available energy, whereas in some reactions that are endergonic in nature, they will require some definite amount of heat for their completion, and such reactions are non-spontaneous and they are endergonic reaction. But the most important thing that you must know is the condition. Just assume A and B are the reactants that will combine in a reversible reaction to give C plus D, which are the products. When this reaction attains equilibrium state at which the concentration of reactants and products will remain same, the Gibbs free energy at this state will be 0. There will be no further increase or release of energy. So the most important thing, if someone asks you if a chemical reaction attains chemical equilibrium, what is the Gibbs free energy at this state? Because at this state, the rate at which the reactants are going to convert into products is equal to the rate at which the products are going to convert into reactant. So there is overall no release or absorbance of energy. That's why Gibbs free energy at chemical equilibrium state is 0. Now we are going to move forward. What is standard Gibbs free energy? It is represented by delta G naught. Whereas if you measure any quantity like enthalpy at standard state, it will also be represented by delta H and over naught, delta E naught. So standard Gibbs free energy is represented by delta G naught. It is defined as it is a free energy change when concentration of reactants and products are 1 mole per liter at standard state. First of all, I want to inform you what is the standard state of biochemical reactions. There are some type of reactions that are happening in your chemistry lab, physical reactions, like if these are gases, ideal gases, they have some specific standard state represented by STP, standard temperature pressure. Whereas standard state for biochemical reactions happening in human body has different conditions or standard state. It does not depend on pressure or temperature, but the standard state for biochemical reactions happening in human body is pH7. So if someone asks you some biochemical reactions, what is the standard state for those biochemical reactions like glycolysis or other biochemical reactions, so it is pH7, and this standard state can be changed according to the conditions, whereas ideal gases, liquids has different standard states that remain same in all the conditions. So first you must know what is the standard state. Now I am going to relate how you can calculate the standard Gibbs free energy and explaining this definition. Delta G, which is a Gibbs free energy, is equal to delta G naught, which is a standard Gibbs free energy, plus R general gas constant, T absolute temperature, I, and this is called natural logarithm into concentration of products over concentration of reactants, and this is the basic relation for calculation of standard Gibbs free energy by using the Gibbs free energy. Like I have explained you, when a chemical reaction is attained chemical equilibrium state, the delta G at this condition is equal to 0, because there is no release or absorbance of energy. So delta G will be 0 when a chemical reaction is present at equilibrium state. But just for explaining this definition, what will happen if the concentration of products and reactants become 1 mole per liter? Because to calculate the standard Gibbs free energy, there are two conditions. One, the concentration of reactants and products should be equal to 1 mole per liter. Second, it should be measured as standard state, which is pH7. So if concentration of products and reactants become 1, then the natural logarithm of 1 will be equal to 0, and when this condition becomes 0, these values, natural logarithm of product over reactant is equal to 0 when the concentration of reactants and products are 1, then this whole value will be equal to 0 and delta G will be equal to delta G naught. So in your Harper's and Biochem Lippincotts, they are explaining this, that standard Gibbs free energy is equal to the Gibbs free energy change when the concentration of reactants and products is equal to 1 mole per liter. So when the concentration of reactants and products become 1 mole, then this whole value become equal to 0 and delta G will be equal to delta G naught. So I hope you have understand the definition of standard Gibbs free energy. Now I am going to calculate the value of delta G naught. So just assume reactants have been converted into products and attain chemical equilibrium, or reactants and products are in chemical equilibrium, and delta G at this state, as I have explained you, will be equal to 0. So left hand side delta G will be equal to 0, is equal to delta G naught plus RT into natural logarithm of products over reactants. Delta G is equal to 0 because the reaction is at chemical equilibrium state and the concentration of products and reactants will be constant, not changing, not equal to each other, as at equilibrium states concentration of products and reactants will be constant but not equal to each other. Delta G naught will be equal to minus RT into natural logarithm of products over concentration of reactants. As chemical K of equilibrium is equal to concentration of products and reactants, you can also replace this value by K of equilibrium, which you have also read in your intermediate classes. So by using this formula you can calculate the standard Gibbs free energy. What thing you must know to calculate this standard Gibbs free energy? You must have to know the concentration of products and reactants at equilibrium state. So if you know the concentration of reactants and products at equilibrium state, you can easily calculate the value of delta G naught for any biochemical reaction. So this is a simple thing. I hope you have understand how you can calculate the standard Gibbs free energy and Gibbs free energy. Now I am going to show you the relation of second law of thermodynamics with Gibbs free energy. Delta G, which is the Gibbs free energy, is equal to delta H enthalpy minus temperature delta S, which is entropy. So this is the relation between the Gibbs free energy and second law of thermodynamics. Second law of thermodynamics states that for those chemical reactions that are spontaneous, mean that they have delta G negative, there will be increase in entropy of the environment. Increase in entropy for those reactions that are spontaneous in nature. This is the second law of thermodynamics. So I am going to explain this, like what second law of thermodynamics want to say. Just assume this is a system in which biochemical reactions or any chemical reactions are happening, and this is surrounding. What is entropy? Entropy is any disordered randomness. It will be positive when heat is released into the surrounding. It means something is going to add into the surrounding and changing or increases the randomness of the environment. So delta S will be positive when the delta G of this reaction is negative. If this is a highly exothermic reaction, then delta S will be highly increased according to the magnitude of this exergonic reaction. So this is the second law of thermodynamics. If you do not know what is happening inside the system, but you know what is happening outside the system, and if entropy of the surrounding is increasing in positive values, R is equal to 0, it means that this reaction is exergonic or spontaneous, maybe endothermic but spontaneous. This reaction entropy will help you in determining whether a reaction is spontaneous or not spontaneous, or if this reaction is endergonic and requires some heat, the heat will be provided by the surrounding and entropy of the surrounding will be negative. So this is the relation of entropy with the Gibbs free energy of biochemical reactions. In next lecture we will discuss the high energy phosphates, low energy phosphates, how ATP transfer energy from one place to other place in cells and significance, many other important concepts. Hope you best of luck. Like, subscribe, share. Thank you. Allah Hafiz.